Hello, and welcome back to For the Love of dPCR for our next session highlighting the use cases and applications of dPCR across a variety of research fields. Before we get started, I want to encourage everyone to take a look and answer our survey questions on the left side of the event window. In today's session, we're honored to have Dr. Andrea Gaedigk and Wendy Wang join us to present their work on CYP2D6 copy number determination using digital PCR. Our first speaker is Dr. Gaedigk, a professor of pediatrics at the Children's Mercy Research Institute in Kansas City, where she directs the pharmacogenetics core laboratory in the Division of Clinical Pharmacology and Therapeutic Innovation. Dr. Gaedik's work focuses on the variation in pharmacogenes in diverse adult and pediatric populations in clinical and basic research, where she has led over 245 peer-reviewed publications. She also is directing the Pharmacogene Variation Consortium and closely works with the Pharmacogenomics Knowledge Base, the Clinical Pharmacogenetics Implementation Consortium, and other organizations and groups to promote the implementation of pharmacogenetics in clinical practice. Dr. Gaedigk is a collaborator on many internal, national, and international research projects. Our second speaker in today's event is Wendy Wang. Wendy is a researcher and laboratory supervisor of the Pharmacogenetics Core Laboratory in the Division of Clinical Pharmacology, Toxicology, and Therapeutic Innovation at the Children's Mercy Research Institute in Kansas City, Missouri. She received her Bachelor's of Science in Clinical Laboratory Science at the University of Kansas Medical Center, concentrating in both clinical laboratory science and molecular biotechnology. For more detailed speaker bios, please read more on the left panel of your event window. We are thrilled to have Wendy and Dr. Gaedigk with us today to share their insights and expertise. Without further ado, let's get started. Dr. Gaedigk, the floor is yours.
Thank you very much, Christina, for the kind introduction. In today's webinar, we will tell you about cytochrome P450 copy number determination and how the absolute qDPCR system is helping doing this. Our objectives today is to give you a brief introduction into pharmacogenetics and individualized drug therapy and an overview of the cytochrome P450 gene, which is a rather complex gene locus. And in the second half, we provide insights into use of digital PCR and how we can use this method to determine CYP2D6 structural variation with the absolute Q system. Many factors impact drug metabolism and response. And as you can see here around the circle, genetics can be a very impactful contribution to determine the variability of an individual patient to drug response. So enzymes that are involved in drug metabolism, transporters, and targets are highly polymorphic, and the promise of how we can optimize drug therapy is to not give every person the same amount and the same drug, but rather select based on a person's pharmacogenetic profile, which drug is the best and which dose is the best. So a slower dose or a higher dose might just be right for a given person. Almost every person has at least one variation in one of the pharmacogenes. And this is just one study that was done in the UK Biobank. And in average, you can find 3 to 4 variants in an actionable pharmacogene. So understanding pharmacogenetic variation is very important for everybody, as at some point in our lives, we are taking some drugs. Important pharmacogenes include cytochrome P450 and there are many clinically used drugs that are metabolized through this enzyme. So as you can see, the top red circle in this spider graph branches out to a whole lot of different drugs and drug classes. And so this particular enzyme metabolizes, for example, antidepressants, antipsychotics, pain medicines, and also drugs used to treat cardiovascular conditions. So it is a very important gene to understand what the function is for drug therapy. When we look at activity of this enzyme across a population, what we can see is there are individuals on the rapid end of the activity distribution. They are on the left in the graph, and those shown in red are those we call poor metabolizers. So these individuals have no or little activity for cytochrome P450-2D6. And this population distribution you see here is for an European population. It can vary depending on what your population background is. So in African American individuals, for example, it is about 5% of individuals who are poor metabolizers, where it is about 7% in Caucasians. And in people of East Asian origin, it's only about 1%, but there are considerably more intermediate metabolizers. And the underlying genetic variants can also dramatically vary. So certain variants we only see in certain populations or at different frequencies, depending on what a person's ancestry is. In pharmacogenetics, we use a nomenclature that uses star designations, as you can see on top on the left. And this particular nomenclature is really just a shorthand for haplotype. And what you see here is both strains of allelic variation, one inherited from the mother, the other from the father. And the ones in color highlighted are those that are important for the function, but there are also many other synonymous SNPs on these two allelic variants. So this just signifies how complex this gene locus is. But on top of just having these single nucleotide variants, there is also copy number variation. There is gene deletions, gene duplications, and multiplications, and what we call hybrid genes, where the CYP2D6 locus rearranged with the pseudogene, the CYP2D7. And so we have hybrids that consist of CYP2D6 and 2D7, and those of 2D7, 2D6, so different structures. And there are all sorts of complex combinations of the ones I have mentioned above. And so all these structural variant or CNV alleles can have increased, normal, decreased, or no activity. So it is therefore utmost important to not only understand what single nucleotide polymorphisms there are in a person, but on top of that also whether there is structural variation and the nature of these structural variants to understand what the phenotype or metabolizer status is of the patient. And in the next slide, this is just an overview to kind of see these structural variants. So on top, in the top portion labeled A, you see the reference gene locus. In the portion labeled B, you see identical gene duplications. So you could have a star 2, for example, duplicated, which constitutes 2 normal function gene copies, but you could also have a duplication of non-functional or decreased ones. And then you can have the deletion. In the bottom portion gives you an idea of all these different complex structures where you have these hybrids indicated by the first 2 lines in the C block here, where you see that the gene starts with a 2D7 portion, then switches over to 2D6. And there are different hybrids where they switch over in different regions. And such hybrids can occur together with fully functional gene copies or other non-functional gene copies. So there's all sorts of complex rearrangements going on. And these are quite complex and challenging to investigate. So there is a number of methods that have traditionally been used, including qualitative long-range PCR, but also sequencing of targeted amplified region, then the more traditional quantitative PCR, droplet digital PCR, and now we are using very successfully the absolute Q system, helping us to characterize the copy number status at different regions. So where do you find all that information about the variation in the gene? There is the database of the Pharmacogene Variation Consortium I'm directing. And here you have access to all the different variant allelic variants we have defined. There is no time on this webinar going through everything that is available in the database, but please check it out. There is a document available that describes structural variation in more detail if you are interested. The PharmVar tutorial on CYP2D6 written by experts is also a great resource to read up on more detail what we have done. And here we provide some examples of copy number testing. And in that top example, you see a diplotype where we do have a duplication of what we call the star 2 allele, which is paired with the star 4. So this is an identical gene duplication. And if you interrogate this gene at 4 different spots, you get a signal at each of these spots. So your copy number call will be 3. So you know there are 3 copies. You do, however, not know what the nature of these allelic variants are. In the bottom example, we have what we call a star 5 gene deletion, which is the top line. And on the bottom, a star 68, we do have a non-identical duplication. The gene shown to the right is all the way through that comes from CYP2D6. So this is why you see 4 of these green arrows indicating you get a signal there if you probe in dPCR. But the other gene to the left mostly contains CYP2D7 derived sequences, and your copy signal only comes from that exon 1 or 5' UTR region. And so together you essentially have a copy number signal for one of these regions of 2, but a copy number signal of only 1 for the other regions. And that difference in the copy number signal tells us there is a hybrid present. So this just gives you an idea of how complex it is and how we use digital PCR. And with this, I will turn over to Wendy, who will walk you through some of the examples in much more detail.
Thanks, Andrea, for the super awesome overview of pharmacogenetics in CYP2D6. It is a super complicated gene, and we're so happy that we have digital PCR to kind of help us figure out what's going on. Before I dive into the project that we were working on, I wanted to explain why we chose digital PCR for copy number determination over other standalone copy number methods. So another method that is typically used for copy number is called the delta-delta CT method, which is usually run, well, not usually, it is run on quantitative PCR, or some people might know it as qPCR. So this method requires comparing your sample to an internal reference gene and then also to other samples of known copy number. And each of these samples, your unknown sample and your DNA standards, need to be run in at least triplicate or quadruplicate for confidence, which is a lot of time, a lot of reagents, a lot of effort, and someone with really, really good pipetting skills. Unfortunately, that can't be everybody. So we can do something a little bit different, a little bit better. And dPCR is different because it doesn't rely on those DNA standards that I just talked about to interpret copy number value. with the application of Poisson statistics to thousands of binary readings of either positive or negative signals, you can quantitate how many copies of DNA there are. So in this example that you see in my little self-made doodle, you have compartments that are filled with different signals. And each of these little lights represents your assay. So the blue is your unknown target that you're wanting to figure out what copies there are, and your green is your reference gene target. So what you're going to do is the instrument is going to read these, and then it's going to use that positive count to approximate the DNA concentration in units of copies per microliter. This in turn gets relatively calculated to the reference, and that is used to determine the copy number value. So like I said, there are a lot of advantages to dPCR. One of those is that you don't need other samples to interpret your copy number. You just use the sample itself, and that gives you your calculated value. And with qPCR, like I said, you have to do triplicates and quadruplicates, not only of your sample, but your DNA standards. So just to figure out one copy number value, you may have to do 9 or more reactions just to get this one little piece of information. And because of all of these things, I think that dPCR is really reliable. It's easy to perform. It's user-friendly. You don't need to be as skilled with the pipette as qPCR, which is very helpful. And additionally, we are able to resolve higher copy number states with digital PCR. With qPCR, typically we can only resolve up to 3 or 4. So with all that hype, let me get into what we actually did. Our goal was to validate CYP2D6 copy number determination for the absolute Q dPCR system. And we split this up into 3 main experiments. So the first one is the one-pot restriction enzyme digestion. The second one is multiplexing different copy number targets. And third is putting it all together and validating this whole workflow with different sample types. And to make sure that we knew what we were doing was correct, we compared our dPCR results to samples that have been extensively characterized qualitatively with long-range PCR, so XL-PCR, TaqMan genotyping, Sanger sequencing, stuff like that. And also we compared our results to our other digital PCR platform, also known as ddPCR. And when we get our copy number values, we wanted to make sure that they are within range of an integer value. So typically our threshold was 0.25 of an integer value. So for example, if you have a calculated copy number value of 1.75 to 2.25, we called that good. It's at 2. The other thing that we looked at to make sure that our samples and our results were consistent is that we try to keep our target concentrations between 100 and 500 copies. This was pretty arbitrarily picked, but we just wanted to make sure like all of our things were consistent. Okay, so for experiment 1, we wanted to validate the one-pot restriction enzyme digestion. Why this is really important to digital PCR and CNV is that you really need a good digestion to chop up all of your parts of your DNA for equal dispersion between your compartments. And traditionally, this is done as a separate step, maybe the day before or an hour before, and then you load it onto your plate, and then you put it on your instrument, then you get your results. So there's kind of like a multiple step process here. What we developed was skipping that first digestion step and just mixing everything together, doing an on-bench incubation and throwing it on the instrument. This was really, really helpful because it's not only saving time and effort and reagents, you're not going to have an extra random little tube of digested DNA lying around that isn't good for much else. So with this setup, we used 2 assays, which is the 5' UTR and the exon 9. And you can see it in my little gene graphic here that's kind of flanking this gene region. And with our reference gene, we are using TERT as our 2-copy control. With this experiment, we picked 8 Coriell's DNA standards of varying copy number states to make sure we're hitting all our different copy numbers. And we picked BGL1 as our restriction enzyme. for the reason that it cuts multiple times within the gene without cutting within the assay region. And here are our results for these 8 samples. I don't know what else to say other than like everything looks good. But just to get you acquainted with these numbers, I decided to make these graphics on the right here. So in blue and orange are visual representations of where these numbers are coming from in the actual gene structure. So this first sample, NA18933, it's one copy all the way across because one of the alleles is completely deleted out. So you only have one copy of CYP2D6. So the red here is the 5' UTR, the green is the exon 9 signal. The next sample in orange is what we would call a copy neutral sample. So it's just a normal one copy of CYP2D6 on each of these alleles. So you get one copy from the one allele and then one copy from the other allele, and that makes 2, and that is for both 5' UTR and exon 9. In these 2 samples here, we're kind of getting different flavors of duplications. So when we're getting 3 copies, we know one of the alleles is duplicated, like you see in the blue. And if one of them is duplicated, it means that there's 3 copies total, and we are getting that correctly reflected in our calculated C and V values. And this 4 is really interesting because you may expect a duplication on each allele to get 4, which is totally possible. But the sample is really cool because we actually see a triplication on one of these alleles, which really tells us that this restriction enzyme digest is working to chop up between all these targets. This next couple of samples is where it gets a little bit complicated with those hybrid genes that Andrea was talking about. So what you see here is a dropout of signal when that gene region has sequence derived from the pseudogene CYP2D7, which I represented in orange. So the sample in blue has 4 copies of 5' UTR because the front part of the gene is still CYP2D6. But when we get to this orange part, which is exon 9, we're not getting signal from those first 2 genes. And so we see a dropout to 2 copies. And when you see an unequal, imbalanced C and V, that is a really good indication that we have some sort of hybrid thing going on. The sample in orange is another hybrid, but it's kind of a different flavor of a hybrid where the front part, 5' UTR, is converted to 2D7, but the back part is okay. So that's how we get a decrease in 1 copy in this orange sample. And these last 2 samples are really interesting. I wanted to emphasize these because even though we get the same copy number for both of these samples, so 3232, you can see in my graphic that I have different amounts of orange. And that is because since we're only interrogating these 2 regions, there's sometimes important information that we are missing out on, like where CYP2D7 is converting. And just to really hit home how important the restriction enzyme digest is, we performed some experiments with some of these selected Correal samples where we didn't digest at all. So we took our copy neutral, the NA17111, which is 2 copies all the way across, didn't digest it. It continued to give us a good, strong 2 copies. But you can see from these samples that I have highlighted in this gray, yellow, and also the red, when you get to higher copy number states, the digestion becomes increasingly important because you need to be able to separate those copies. You see a decrease in the calculated copy number value. So the number is supposed to be as close to 3 as possible, but you can see 2.58 and 2.65 as falling outside of that 2.25 threshold that we set for ourselves. And when you look at the number in red here, you may think just without any other context, like, oh, it's within that 2.25, that's a copy number of 3. Well, that's actually wrong because what we expected is actually 4. And the reason why this is really important to understand, because if you are not separating your copies, what's happening is that these gene locuses are co-localizing together in the same compartment. So you have a decrease of signal to actual target, and that's why you get a decrease of that calculated copy number value. And it's wrong, so we don't want that. So moving on to experiment 2, this one really focused on multiplexing multiple CYP2D6 target regions. You can do this a couple of different ways. So on our ddPCR platform, we multiplex by what we call we multiplex by amplitude. And what happens is that if you have a instrument that is a 2-channel fluorescent instrument, you usually put your reference assay on one channel and your target assay on the other. So naturally you would think that that is only a 1 target assay, but what you can do is add another assay of the same fluor color, but change the concentration. So you get this discrimination of different clusters based on what is present in each of these droplets. So my coworker and I like to joke it's about reading the prophecy in the blobs, and that's what it really feels like, is understanding how all these clusters work. Alternatively, what you can do is multiplexing with optical channels. So the absolute key is really awesome because it has 4 optical channels that we can play around with CNV. So we have our reference gene on one of them, and then 3 other ones for unknown targets. And that's what we did with this experiment. So we put 5' UTR, intron 6, and exon 9 on each of those optical channels, and used we tested both TERT as a reference gene and also RNase-P as a reference gene. So the experimental setup is the same as experiment 1, but we just added the intron 6. And here is a visual of where these targets are hitting, just so you can have an idea of these regions. I think it's important to be able to visualize what is happening in these structures because it helps you understand what is going on in the gene. And here are our results. Again, I know it's a lot of numbers, but I hope you can trust me when I say like everything looks good, everything looks great. With both of the reference genes and everything that is consistent with our previous testing. But the 2 samples I wanted to point out is the samples that I talked about last, where we have the 5' UTR and exon 9 samples where their results were the same, but where they switched over to 2D7 was different. Well, with the addition of intron 6, you get more of that information. You can see, okay, the sample in blue, only exon 9 is 2D7, so you're getting that extra signal, the 3 signal, from the intron 6. Whereas the sample in orange is a little bit of the opposite, where it switches over to 2D7 much earlier before intron 6. So with that additional target, you're capturing more information, which gives you more data to describe these gene copies. So finally, for experiment 3, we're putting everything together and testing it on different sample types. And this setup is basically the same exact one as the previous one. We're using our 3 favorite CYP2D6 regions, 5' UTR, intron 6, and exon 9. The only difference is just the sources of the sample. So previously, we were only using Correal samples. We have 4 additional ones that we added to this set, but we also looked at DNA that was sourced from blood, saliva, and liver tissue. And with the reference gene, we defaulted to TERT, but if anything strange came up, we followed up with RNase-P as well. So with the Correals, these samples we picked for a reason, and I included their consensus genotyping in the slide, not to hopefully confuse you, but just to help you appreciate how complex these structures are. I also made a little cheat sheet table, so if you wanted to pause this and try to figure out where these copies are coming from, you could. But the overall picture I wanted to paint is that CYP2D6 is really complicated, and dPCR has really helped us to get some pertinent information to describe these structures. We were able to resolve some pretty high copy number states of 5 and 6. And the other sample I wanted to talk about was this NA19317, which is a *5*5, which describes a deletion on both alleles. And you can see that our expected matches are observed. This 0 all the way across the board is really important in giving us evidence that these assays are gene specific. So 2D6 and 2D7 are very, very similar. And with this 0 copy all the way across, you know your assay is 2D6 specific and not carrying signal over from 2D7, which is what we want to see. The table that I have set up here is just selected samples from what we all tested. Again, like everything looks bright, shiny, and awesome. For liver, blood, and saliva, we were able to capture copy number states between 0 and 4 for this set, and all of our testing was consistent with the diplotypes and all of the previous testing that we did with it. It even outperformed our ddPCR run in some of these samples, which I thought was pretty interesting. And just for fun, it's not close to Christmas, but we can pretend that it is. We had some bonus findings, yay! So we found copy numbers in reference gene, which copy numbers in a reference gene, which is really interesting because you would expect these to be copy stable. So I'm going to tell you about the sample. The sample is H20139, and when we ran it through our method, we were getting these really off calculated copy number values. So you can see that they're pretty much close to 1.5 all the way down. And because these were off, we ended up retesting it, but with a different reference gene, RNase-P. And with that, you can see how it pretty much resolved the issue. And we were trying to hypothesize why this is. And what came up was that maybe there is a copy number variation in the reference gene. So after finagling around some of these assays, we were able to detect the copy number in TERT. So we made TERT the target and using RNase-P as the reference for this other assay. And here you can see with TERT, we have 3 copies of that target using the RNase-P as the reference. So really interesting sample that we have here. In conclusion, we validated the 1-pot. Everything looks great. We validated the CYP2D6 triplex of 5' UTR, intron 6, and exon 9 together. We were able to resolve copy numbers between 0 and 6, and the sample types that we tested were corneal, blood, saliva, and liver, and all of our copy numbers were consistent with what we had previously observed. Some takeaways that I wanted to bring up is that the 1-pot digestion and the multiplexing methodologies that we incorporated make the workflow super easy and really efficient to do. And, that's always beneficial. More data with less time and less effort is always awesome. And also I wanted to bring up the very, very rare chances that you may get copy number variation in your reference gene. So when you see copy number variations that are off, maybe consider that as one of your troubleshooting steps. And finally, some other things that we were working on with dPCR that we're really excited about is doing copy number in other genes. DPYD, I think, is actually published right now, so you can give that a little googly goo if you want to. 2A6 is another gene, so if you think 2D6 is a beast, 2A6 is a whole other monster. Yeah, that's going to be rough, but we're doing okay. The other applications that we're looking at is allele-specific applications. So with this project, we were looking at copy numbers of the genes, but with the allele-specific applications, we're looking at copy numbers of what variants are present. So this helps in better characterizing these duplication, hybrid, and tandem structures. And finally, we are also looking at higher level multiplexing as well. So that'll be really exciting to be able to combine more information into one reaction. And I just wanted to say thank you, everybody, for listening to our talk, and we will be taking questions.
Thank you, Wendy and Dr. Gaedigk, for a really incredible presentation. I'm really excited to start answering some of the questions that have been populating the chat. So I'll go ahead and take a look now. So one of the first questions that we have is if genotyping is not assessing copy number variation accurately, what does that mean for a patient's phenotype prediction?
Well, that is a really good question. So if a person has 2 copies of the gene and then another copy of a functional gene, that would make that person an ultra-rapid metabolizer, and meaning that this person is metabolizing drugs much faster and might need a higher dose or should go to a different drug regimen. If that is not known, that person would be classified as a normal metabolizer. And so it is really important to understand what these copy number states are to accurately determine what a person's metabolizer status is. And so that adds another layer of complexity of testing, but it is very important for the accuracy of the phenotype prediction. And this is just the basis on which all these clinical prescribing recommendations are made.
Great. And then maybe a follow-up question to that I see here is, understanding some of the features of digital PCR that make it really advantageous. Is it the additional precision in that readout? Is it the speed, or is it kind of both that kind of support that overall advantage?
Yeah, so I would definitely say it's both, because dPCR is such a huge leap from qPCR that it is so much more precise, it is reproducible, that there are a lot of things that you don't have to worry about with dPCR than with qPCR. And additionally, because it is so user friendly, it's really easy to train people on. I picked it up pretty quickly and didn't have to fuss with getting this technique right, left or right. I think all of it was really, really great.
Fantastic. So another question we have here is, early in the presentation, we saw a lot of complexity around copy number states. Why do you see dPCR to be beneficial over a technology like sequencing?
Well, sequencing, the question for sequencing is really a complex one. So if you think of Sanger sequencing, you first need to know whether there is a copy number state present before you can then amplify fragments to sequence. So Sanger sequencing itself does not give you a copy number state. When we talk whole exome sequencing, this is really also very difficult just to determine copy number due to the nature of exome sequencing. Now, when we talk whole genome sequencing, that has really become much better along the development of bioinformatic tools to call these. However, it's still not as perfect as we want it to be, especially if we are thinking clinical applications. And so ultimately, it's probably long read sequencing that is a little bit the holy grail to also detect and characterize these different structural variants in 2D6 much more accurately. On the other hand, we still need to really see how good these sequencing methods are to determine higher copy number states. And I think this is where digital PCR still has a great advantage.
Yeah, so it feels like oh, sorry, I was going to say, it seems like
so with these technologies that do CMV on top of genotyping or sequencing, the CMV is integrated into the platform, right? A lot of these may be financially prohibitive, but if you have a platform that just does genotyping, having something like dPCR complement it is going to be a lot more financially suitable. So yes, you can get CMV from other platforms, but you have to pick the one that makes the most sense for the environment that you're in.
Yeah, fantastic. Of tools in your toolbox kind of scenario. So another question we have that actually gets more into the data analysis perspective is really what type of data analysis is involved for copy number determination by dPCR, and is it more or less complicated than what we'd typically be familiar with for something like qPCR?
So the data analysis is actually really easy. There isn't much at all. You just kind of have to make sure your clusters are separated, and the software itself just spits out a calculated CMV value. You don't have to do much interpretation to get a copy number value. As opposed to qPCR, what you have to do with the software is a bunch of calibrations with that delta-delta CT method that I talked about, because you're comparing a thing to then compare it to other things, and you can do that manually and a spreadsheet application too, but you do have to have training to understand how to interpret the CMV.
Fantastic. So another question we have coming in is on the use of endogenous controls. Is there any benefit to using more than one endogenous control in an experiment like this?
Absolutely. So with the reference genes that you use, there's always going to be a non-zero chance of having a SNP underneath the assay that may affect the way it amplifies. So doubling up on your reference genes is just going to lower that probability that something weird is happening in one of them, and you can default to another one. And especially if you're doing like higher plex multiplexing, you're getting a lot of information in one reaction. Like it would be really sad if you have to redo a reaction just to repeat it with a different reference. So if you can do 2 in one reaction, then you don't have to repeat it if something is weird in one.
Fantastic. And so it looks like we have time for one more question. With respect to the additional sample types that you tested, you mentioned that some retesting was needed. How often did you find that you needed to do that retesting?
It kind of depended. So I didn't go much into it in the webinar because we just had so much to talk about already, but we did have quite a few samples. I don't want to say quite a few, like 2 or 3 like samples that we really had to follow up on. And it really came down to sample quality. Like one of the interesting ones that we talked about in our paper, we tested by following up with a different reference gene. We made dilutions to make sure there wasn't any inhibitor issues. And like what happened was we just picked another extraction of the same sample where the quality was much better. So we ran it out on a gel to see how intact the DNA was. The original aliquot that we had used was all messy and degraded and really gross to look at. And the other one was nice and compact and a higher molecular weight. So a saying that we love to throw around in our lab is garbage in, garbage out. So, there's only a certain amount that you can do with a bad DNA sample. So that's a really good place to start. But beyond that, like I found dPCR to be very robust in dealing with things that may affect amplification. Like it's more accommodating than other things are.
Yeah, I mean, this is the same with any workflow. So if you make sure you have good DNA preparations to start with, I don't really see a lot of issues really coming up that you would need to repeat a lot of samples unless, it's really an operator error at some point.
It happens.
Yep, I know.
All right, well, that is all the time we had for questions today. I want to thank you both again for joining us for this incredible presentation, and thank you to all of our audience members for their great questions. This will be available on demand, so I encourage you, if you missed out or if any of your colleagues missed out, please share the link and feel free to watch again at your own convenience. But thank you again, and I hope you all have a wonderful rest of your day.
Well, thank you for having us.
Thanks.
Bye.
Thank you again for joining us to learn more about copy number determination by digital PCR. Before we end the event, I want to encourage everyone to take a look and answer our survey questions on the left side of the event window, and to introduce a new product to our digital PCR portfolio that can help you explore new applications and research possibilities. For our survey, the first question is, I use or am interested in leveraging dPCR for the following applications: absolute quantification, standards or control material quantification, gene expression, copy number variation, viral vector quantification, or gene editing confirmation. Please answer the question in the panel on the left. For our second event question, the question is, I am aware of the new Applied Biosystems Absolute Q Universal DNA dPCR Master Mix, which unlocks a suite of pre-designed TaqMan assays for use on dPCR. The answers are yes or no. And also go ahead and fill that answer in on the left-hand side of your screen. If you answered no to question 2, that's completely okay. We're very excited to introduce a new product to our digital PCR portfolio that will help you to explore new applications with ease. So we're introducing the Absolute Q Universal DNA Digital PCR Master Mix, which was overall designed to lower the barrier to entry for digital PCR by providing you with access to over 20 million pre-designed TaqMan assays. With an extensive assay library and a performance guarantee with broad compatibility across a range of applications, you can easily complement your existing qPCR workflow with a cohesive Applied Biosystems TaqMan experience. This is all a part of our QuantStudio Absolute Q Digital PCR Solutions family. So we encourage you to take a look at what we have to offer and get in contact with a specialist so that we can help design a dPCR solution for you. Thank you again for attending the webinar. Have a wonderful day.